Okay, so now we'll start the class. So I will speak a little slowly today because I'm not very well. My voice, I'm not able to speak also a lot. So uh, I hope you guys will understand and bear with me. So today, sorry. Hmm. So today we have to study the different kinds of RNAs that are there, the processing of different kinds of RNAs, and the import and the export of different kinds of RNAs, and the function of different varieties of RNAs. So we have studied that RNAs can be mRNA, rRNA, or tRNA. Okay, this all we know. But there are also other non-coding RNAs. So non-coding RNAs are the RNAs which does not code for protein. Okay, so which does not gives an amino acid which does not code for a protein. Those RNAs are known as non-coding RNAs, and these RNAs have a lot of important function. For example, these RNAs are required for the post-transcriptional processing such as splicing. Excuse me. And also these RNAs are non-coding RNAs are also responsible for the regulation of the transcriptional activity of RNA by limiting the production of proteins from the existing RNA molecules. So those small non-coding RNA molecules have a lot of other functions as well. So today we have a lot to cover. So we won't go in a lot of details of everything because then we won't be able to finish it. But for your understanding and for your learning sake, I have put the screenshots of uh, b- uh, some of the important topics or some of the important theory that was there in Sir's class, so that even after the class you can go through it and you can just have an idea. Other things are Some things are self explanatory, so wherever there is a concept building topic, then I'll try to explain the concept, and then whatever else is self explanatory, I have put the screenshot so they can read it later on. Okay, so we'll start with lecture first. Excuse me, just a minute. Okay. Okay, so in the last class we have studied about the mechanisms of intron loss. So today we have to see the mechanism of intron gain. So first we have to study about the introns and their link to splicing. So lecture 1 is about introns and their link to splicing. So in this first we'll be seeing that how introns are gained and lost, how the introns are lost, that we have already seen in the last week's class, if you remember. So you can see over here, just a minute, I think someone has to join. Okay, now you can see over here. See, I'll just yeah. Hmm. So this is your axon and this is your another axon, and in between there is intron. So first this intron is spliced out. Okay, now this spliced out intron can insert into another mRNA where there is no intron. This is your gene. This is a complete exon, you can see there are no introns, so this is your intron less site. Okay, now this exon can be cutted, and then this intron can be inserted into this, and then when reverse transcription happens, then this RNA is converted into cDNA, And then you can see this is a complete DNA which is now made, where there is now intron there. So here we're talking about mechanism of intron gain. So how a particular gene gains an intron, how a particular gene gets an additional intron. So the intron which is deleted from one exon, it can be, it can come from there and it can join to another exon where there are no introns, which is an intronless exons. Means if this is your exon, this is your intron, and this is your exon, exon 1, exon 2, and this is your intron, and this is your another exon 3. So what will happen? This intron would be spliced out from this, and this would be inserted here. So now it will become like this. This exon 3 is divided into in such a way. Let's say it is as exon 3A, exon 3B. And now this intron is inserted. So this is the mechanism of intron gain. Another thing which can help in the gain of electron is transposons. So we have already studied about the transposons in our earlier, I think in the last class only we discussed one question regarding the transposons. So you can see what can happen, that transposomes themselves can be inserted. This is a transposon, and this transposon can be inserted, and then this transposon can be converted into a spliceosomal intron. That means transposomes are not helping in the intron insertion. Transposomes itself are getting inserted. Okay. Do you understand the difference? Transposomes do not help in the insertion of some existing introns. Transposomes themselves gets inserted and then they are converted into introns, spliceosomal introns. Okay, that is what they are saying. Now, how these transposomes can be inserted and how these transposomes can make a cut, because transposomes they have inverted repeats. So at both the ends, transposomes have inverted repeats which help in the recombination, and they have an enzyme which is known as transposase, which helps in the copy paste or cut paste mechanism of transposomes. So this is the mechanism of intron gain. Intron can be transpositioned from one exon to another exon, or a transposon can be inserted and then transposome can be converted into a spliceosomal intron. Then we comes to, then again, then now what is there? This is one thing. Another thing is that intron can be transferred into a paralog of itself. If this is a gene, intron can be transferred into a paralogous gene. I mean, if intron is getting cut from one side, it can be transferred to any other side, okay, where it can recombine. The only criteria for an intron to join to another particular region is that it should have some sites by which it can homologously recombine. For example, what I'm trying to say over here is, if this is your exon 1, exon 2, and this is your intron. Now this is spliced out. Now this spliced out intron can be inserted into any other region. The only criteria for its insertion is that other region should have sites so that it can homologously combine. So it can either insert into a similar kind of gene, or it can also insert to a paralogous gene. So if this is a paralog, this is your exon with the intron, you can see this intron will loop out this these two genes, because they are paralogous genes, they have sites, so they will homologously recombine, and now this intron is cut from here and it is transferred to the other paralogous type, I mean to the other paralogous gene. So this is another mechanism of intron gain. And then self splicing means, self splicing type 2 intron means that we have already studied that there are different classes of introns. Some are self splicing introns and some are introns which are required in the spliceosomal complex. Self splicing introns means that they do not require a spliceosomal complex or anything. They are fully they are fully sufficient. They do not need anything from outside. So what can happen, that self splicing introns, they can be cutted and they can get ligated to some other gene, and then they are converted into spliceosomal intron. That means there is a conversion of self splicing intron to a spliceosomal intron. Okay, so up till now we have studied there are four mechanisms of intron gain. First is intron transposition. Trans means different, position means to position. That means intron is getting positioned from one exon to another exon. Second is transposon insertion. That means transposon is getting inserted between the exon, and then transposome is converted into a spliceosomal intron. Third is intron is getting transferred to a paralogous gene through homologous recombination. Fourth is a self-splicing intron is converted into a spliceosomal intron. So there are, we have studied, there are थोड़ा धीमे धीमे करोगे, बहुत ज्यादा आवाज आ रही है. There are four mechanisms of intron gain. Four mechanisms. Intron gain that we have studied. Now comes what are paralogs. So I am giving a little bit more time on these slides because there are, see, we have 5 classes for this week, and a lot of things are just you have to go through and you will understand. There is nothing much to explain. You just have to read the content. Only some of the portions are there that need some kind of explanation. So I will try to cover some of those topics. Obviously I cannot cover all the 5 topics in depth in this 2 hours class. So I will suggest that you also go through the chapter, and if you have any doubt you can ask me in the subsequent class. Now comes the other important point, that is what are paralogs. Okay, so for this I'll go to the next slide. Okay. Can anybody tell me what are homologous genes, or what is the meaning of homologs? Any general definition of what homologous means, or what are homologs? Can
Same
anybody tell
alleles,
me? Yes?
same alleles are present.
Same?
Alleles are present.
Okay. Any other one? Any other person who wants to try? What are homologs? Anything that you have read in your class or anywhere? Homologous gene, homologs.
Similar gene, similar
Yes.
gene,
So can you be a little more? I mean, I'm not able to hear you. Can you just increase your mic, maybe?
similar gene,
Similar gene.
similar.
Okay. 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 Fine. I could
Ma'am, if
hear
they
you.
have derived from the same ancestral gene,
Hmm.
derived from
Then
same origin,
Okay. So all of you have kind of tried to explain, and I appreciate that you all at least tried and tried to participate in the class. So, and I think Jyoti has answered it precisely. All the other points are also correct only, but when we talk about homologous, the first thing that comes to our mind is common ancestry. That means, excuse, excuse me. That means somewhere or the other they have a common ancestor. They have a connection. They have a connecting point. Maybe that connecting point has before 100 generation, doesn't matter, but they have a common ancestry. That is your homologs. Now, homologous genes are divided into two types. First one is orthologous, or not just genes, just in term, if we talk about homologs, it's not always the gene. Homologs can be your pair of organs or pair of anything. Homologs can be anything, not just genes. So homologs are divided into two types. The first one is orthologs and the other one is paralogs. Okay, so homologs in due course of evolution have separated into two ways. homologs have been separated in due course of evolution into orthologs and paralog. Okay, so orthologs and paralogs means there is one which is separated by speciation events and the other which are separated by gene duplication events. So the one that has been separated by speciation event, that has now become two separate species, they are known as orthologs. And the one that are separated by gene duplication events, they are known as paralogs. That means there is a one very good diagram also inserts this thing, I think lecture. You can go through it also, that if there is a one gene, suppose 1, it got duplicated, it has become 1a and 1b. Now some speciation event has happened. Now how this gene can become two different species? So if you have studied something about if you have a little bit knowledge about ecology and evolution, then there are many different reasons then why a particular species is emerges out into a new species, because of some geographical distributions, that if a particular species has, say, a particular kind of species has three organisms, two of are living here and one of them is living here. Now there has been some geographical event because of which these two got separated. Now it got into some other place, it got into some other place. Now these two cannot mate with each other. Now they both are living in different geographical conditions. Here the conditions are very dry, like desert conditions. So now this species, over the time to survive and to adapt with the environment, this species, suppose these are plant species, let's say these are plant species, so this species will over the period of time will adapt and change itself in such a way that it can retain more and more amount of water. So it will have uh, thorns in them, the leaves would be modified into thorns, and they will be succulents, they will have a lot of water retention capacity in them, and these are living in the normal conditions. After thousands of years, this will emerge out into a completely different species, and this would be a different species. So specialization events means any kind of geographical distribution, or any kind of any kind of calamity, something has happened that has now restricted them to breed with themselves, and they have put them in different kind of geographical landscapes. So like this, some specialization event has happened, and now this has emerged into a different species, and this has emerged into a different species. Then again some gene duplication events have happened, and gene duplication events also leads to some different forms of the same genes. So if speciation event has happened, then that leads, then these two would be called as orthologs because of the speciation events that has happened, which has led them to be different from each other. If gene duplication events leads any difference, those are known as paralogs. This is your orthologs and paralogs. Now, so first thing what we have studied, our first lecture is introns and link to splicing. So first thing that we have studied in the last class, we have studied about intron loss, how introns can be taken out from one particular exon. Here we are studying about how introns can be gained. So there are four mechanisms: transposition, transposome insertion, intron transfer, and self-splicing, means self-splicing introns converted into spliceosome introns. And here we also studied about orthologs and paralogs. Next thing that we have to study about regulation of RNA splicing. So till now we have studied about RNA splicing events. Just a minute. Yes. So till now we have studied about RNA splicing events. So RNA splicing is a kind of post-transcriptional modification that converts your primary mRNA into a mature mRNA. Okay, that is fine. Now, what if there are some problems that happens during this splicing, or what if there are some problems that can lead to an inefficient or improper splicing, then that could ultimately lead to mutations. And if that can lead to mutation, that can ultimately lead to any kinds of diseases. If splicing is not proper, or if the splicing is incomplete. So in both the cases, there would be some kind of pathological or morphological, there can be some morphological or pathological manifestations in the form of diseases. So that is why in our body, the mRNA splicing, sorry, RNA splicing is very well regulated, by some kinds of modification that happens after RNA splicing or during RNA splicing, that regulates the process of RNA splicing. What are those modifications? It can either be phosphorylations, acetylation, hydroxylation, or ubiquitination. So these are the modifications that happens that in a way regulate the RNA splicing event, so that RNA splicing can occur in a well coordinated manner. Okay, just like DNA replication also, we have if you have studied that there are many kind of DNA damages that can happen, like through UV radiations and all, and those DNA damage, we have several repair pathways also, if you have studied about some repair pathway. So just like DNA repair pathways are there, and in cell cycle there are various checkpoints. So the DNA, when it is replicating, it has to pass through all the checkpoint, G1 to S checkpoint, M checkpoint. So these all checkpoints the DNA has to clear, then only it will pass. Similarly, splicing also needs to be regulated so that there are no imperfections in our body which can ultimately manifest in the form of any kinds of diseases. So so post transcriptional, so these are some of the modifications. You just have to read learn I mean know about the names. And then we have also studied that the splicing that happens, it can also be regulated with the help of major spliceosome and minor spliceosome. So you have to understand over here is that mostly splicing happens with, see, I have already told in the last class that both major spliceosome and minor spliceosome, major splicing or minor splicing, they both have same function. It's just that by default, major spliceosome works for splicing, which is U2, which involves U2 type of introns, and in some cases, minor spliceosome comes into action. So now you can understand, just like suppose I have a goal in my mind. Okay, I want to achieve a certain thing. So I want to achieve, suppose you guys have a goal in your mind that you want to excel in your this NPTEL course, and you want to get first rank or whatever, perfect marks or very good marks in your in this course. So you will have that goal in your mind, but then you will also have a plan B. We all must have a plan B, that if not this, then we'll do something else. Then we'll go and then we'll again read, and then we'll again appear for the course, and then we'll again get the good marks. So we always have a plan B in our mind for whatever goal we have desired of. Similar way, our body is also very intelligent, our body also knows that even though we have a very organized and a complex machinery to achieve each and every process, still there are chances that some mishaps can happen. Still, there are chances that some kind of external environmental factors or autoimmune generated factors can uh, can cause a problem in the normal regulation and functioning of the cell. So our body also has a plan B for each and every process that happens inside the body. And these minor spliceosomes are nothing but a plan B for the splicing events to take place. Okay, so if there is some problem in the major spliceosome complex, then U12 type of introns comes into action. first. And also in some cases where major spliceosome is not that prominent, then also minor spliceosome can come into action. But majority, you understand that minor spliceosomes is like a plan B. Okay, so what I'm trying to tell is that RNA splicing is regulated, and this major spliceosome and minor spliceosomes are also a kind of manner to regulate the RNA splicing event. What you have to understand over here is the difference between major and minor splicing. I'm sorry, I'm actually not very well, so you might face this. Okay. So now, major spliceosome has U2 type of introns, whereas minor spliceosome has U12 type of introns. Okay, and you can see that here it has to be AG, here it can be A or G, here it can be C or G. I mean AUGC, that we have studied AUGC, or the 5 prime splice site or 3 prime splice site that we say in your major spliceosome complex, that that consensus sequences are very much conserved in the major spliceosome complex, but that are not as much conserved in the minor spliceosome complex. That is what they are trying to tell with the help of these sequences. Then just as I told you, that if splicing, just like in any process, if DNA replication is happening and there is some problem in DNA replication, it might lead to cancer and many other kinds of diseases. So any molecular process, any molecular phenomena, if it gets any kinds of defects or disruptions, it will lead to some kind of pathological conditions. So similarly, splicing will all, if there are some defects in splicing, it can also lead to different kinds of pathological conditions. Now those mutations can be either in splicing factors or in the splicing pattern. Okay, if there is some mutation in splicing factors, it can lead to spinal muscular atrophy, pigmentosa, myotonic dystrophy. If there is mutation in splicing factors, and there is mutations which affects the splicing pattern, both are different things, you should pay attention to it. Hmm. Then it can lead to beta thalassemia, muscular dystrophy, cystic fibrosis, Frazier syndrome, dementia, and Parkinson. Okay. Then we come to next lecture. So today what we'll do is that, because the questions are very much connected, so first I'll explain you the two lectures, and then we'll solve some problems, and then again I'll explain you the other lectures, and then we'll try to solve some problems. So up till now, are things clear to you? Do you have any, if you have any problem, please ask me. Are things clear? Are you able to understand?
Yes.
Okay, fine. And I can see the attendance is going very exponentially. The attendance is decreasing. Why so? Is it boring, or you don't want to attend the classes? I would suggest you to attend the online classes, and if you have your connections with your other course mates also, then please tell them to attend the classes, because ultimately these also counts in your marks, and not just about the marks, it's more about the knowledge. Or even if you achieve achieving or getting 0.1% from any of the class, then it's more than enough. Anyways, because if I am coming and explaining, even I'm not so well, then I think you guys only have to listen, you don't have to speak. I am the one who has to speak for 2 hours, even I'm not well. So if I am putting that much of effort, that from all my busy schedule, if I am putting some effort to first go through all the lectures, I also go through all the lectures, whatever lectures are given to you. First I see all the lectures, and then I make the PPT, and then I come here to explain. So I have to first read everything, because I also don't know everything. This is also some of the things are also new to me also. So if I can put that much of effort for you, otherwise I could have just taken some questions and I would have just solved them. That would have completed my this thing. I only have to solve questions, make you solve questions. I don't have to explain you the theory part. That is not my assignment. I'm only told to explain the questions, that's it. But I'm putting extra effort and I'm telling you theory so that would be easier for you to understand and you will at least gain something. Just solving 10 questions without knowing anything about the theory does not solve the problem. So if I am putting that much of effort, I would at least expect you to at least come for the class and listen. Okay. So now we come to the next lecture, which is RNA helicase. So helicase in general, can you tell me what is the function of helicase in general? In replication also we have studied about. Yes.
Unwinding.
Unwinding. So
Unbinding
helicase
of
as,
double strands into single strands.
yes. So Kavita and Tarikshwari, I'm sorry if I'm pronouncing your name incorrectly.
Tarkeshwari.
Can you repeat?
Tarkeshwari.
Tarkeshwari. Okay. So Tarkeshwari and Kavita, you both have told the correct answer only. That helicase means that it is unwinding, and when it unwinds, it also kinds of helps in uh, creating a single stranded copies. So we have also studied about helicases. Excuse me. So we have also studied about helicases. In, if you have studied about DNA replication, that before the DNA replication, how the two, this is your DNA. So these how the two DNA strands first unwinds themselves, then how they are sorry, first unwinds and then helicase kinds of cut them so that it can be separated. So all these things we have studied in DNA replication also. So in RNA helicase also has some kind of similar function only. So basically these are RNA dependent NTPase or RNA unwindase. What it does is that it hydrolyzes the nucleotide triphosphate, that is ATP. So it hydrolyzes ATP, and then with the help of that ATP, the energy that it gets from ATP, it performs the unwinding function. That means it needs the energy of ATP. It does its work in ATP dependent manner. It needs the energy, yeah. It needs the energy from ATP to perform its function. So first helicase hydrolyzes the NTPs, means nucleotide triphosphates. So mostly these nucleotide triphosphates are the ATP. So first it hydrolyzes NTPs or ATP, and then with that energy, it acts on the substrate. Substrate can be your double-stranded RNA also, or it can be your RNA protein complexes also. Here in this case, we are not talking about DNA. Our focus is on RNA. Okay, someone wants to join the call. Just a minute. Substrate can be double-stranded DNA also, or it can be RNA protein complexes also. So you can see that there are different types of helicases, and these different types of helicases have different domains. So different helicases have different domains. So the difference in the different uh, varieties are because they have domain difference. Okay, these are the different kinds of helicase. You can just go through it. This is just like to read. Some has dead box protein, such another one is deck box protein, skip 2 family. So these are different kinds of helicases. This is the whole RNA helicase family. So you can see a typical structure of RNA helicase. It has an ATPase domain. That means a domain which is used to hydrolyze ATP to get the energy. Then the helicase domain, which is used for unwinding, and then a domain which is used for interaction with the RNA, whose RNA has to unwind. So this portion will interact with the RNA which has to be unwinded. This portion will hydrolyze ATP to generate energy, and this portion will actually cut. So these are three main portions in a typical RNA helicase family. So just like what we said, that the substrate, it can either be double stranded RNA or RNA protein complexes, and 7 conserved motifs for helicase family. 1 2 3 4 5 6 7 8. Okay, so maybe yeah, you can see these motifs are conserved. Some conserved motifs are there, and actually the image is not very clear, but I think that the domain which is responsible for interaction, that is different. And one, okay, okay, these are the okay, these are same only, 1 1a, 2 3 4 5 6, and this is the 7. Okay, so basically typically each and every RNA helicase will have 7 conserved domains. Okay, what I'm trying to say is that all the domains are not conserved within, I mean, whatever domain is there in RNA helicase family 1, it will not be the same in here, but for dead box protein this will only be the domain always. Do you understand what I'm trying to say? I'm trying to say that this particular domain is not conserved across the RNA helicase family, but within this family it is always conserved. That ATPase domain for dead box protein will always be this only. It will not be anything else. For dead box protein, it will be always be this. For skip2 family, it will always be this. That is the conserved domain means here. Then now, so now right till now we have studied about RNA helicase. Now we'll see the RNA helicase function. What is the function of this RNA helicase? So basically these RNA helicases are used for ribosome biogenesis. Nothing to explain much, you just have to read that these RNA helicase basically help in intra and inter RNA interactions, and they also mediate help as in for cutting the intra and inter RNA interactions, and they also unwind RNA protein interactions. So basically in ribosome biogenesis also RNA helicase has a role, and this role is achieved by these many achieved by performing these many activities, that it can unwind intra and inter RNA interactions. So what is the difference between intra and inter? Can anybody tell me? Very basic. Intra RNA, if I say intra RNA interactions and inter RNA interactions, what is the difference between the two?
Two different RNA and the same RNA.
So within the same RNA, that would be inter or intra.
Intra.
Intra. So within the same RNA, it would be intra, and between two different RNAs, that would be inter. Just like we have inter-school competitions, inter-college competition, that means between two schools. And intra means within. Okay. Next is now. Now we see what are the various molecular biology processes where RNA helicases are required. Okay, till now we have studied what are RNA helicases, how they perform, how they function. So RNA helicase basically require the function, require ATP. It hydrolyzes ATP, and then with that energy it acts on its substrate. Its substrate can either be a double stranded RNA, or it can be RNA protein complexes, anything it can be. Then there are different RNA helicase family, and each RNA helicase family has an ATPase domain, helicase domain, and RNA interaction domain. Now we'll see what is the function of RNA helicase. So RNA helicase is required for no, I think what I need is a highlighter. So RNA helicase is required for pre it is required for pre-mRNA splicing. The first function of RNA helicase is pre-mRNA splicing. So this is your exon, intron is there, and this is the whole function of uh, splicing that happens. So in this splicing event, more than 8 RNA helicase participate at various steps of RNA splicing. You can see these are the RNA helicases, the yellow ones. And helicases help in unwinding of RNA and RNA protein complexes. So these U2, U1, these are all protein complexes. No, when it comes with protein SNRNP, when it forms SNRNP, SNRNPs, these are small nuclear RNA plus protein. These are protein complexes. So basically, these RNA helicases, they are involved in mRNA splicing, and more than 8 RNA helicase participates at various steps of splicing. So first thing that we got to know is that RNA helicase are required during splicing. Then they are also required during RNA maturation. They are required in RNA export. They are required in translation. And then they are also required in, yeah, so these are essentially these are the important functions of RNA helicase in pre-mRNA splicing, in RNA maturation, in RNA export, and RNA translation. So these are the various four processes, molecular biology processes, where RNA helicases are involved. Now in detail we'll see that how RNA helicases are involved in RNA export and in translation. So you can see that these RNA helicase, basically they kind of maintain the correct conformation of RNA protein complex for the export through nuclear pore complex. So basically when these RNA helicase, they act on RNA protein complex, okay, they act on RNA protein complex and they maintain its correct conformation. First thing. Second thing is they are involved for unpacking, because when they unwind, they kind of unpack it also. So they help in unpacking of the RNA protein complexes. Then they are used for removal of shuttling proteins. They remove some proteins also, and they are also required for uh, translation that we are going to study here. Then so these are the points for which are required to be maintained for the export of RNA. The RNA protein conformation has to be correct. RNA protein has to be unpacked, and the other extra proteins, shuttling proteins. So what are shuttling proteins? Can you tell me what is the meaning of shuttling proteins? Anybody? Anybody knows the meaning of shuttling proteins? No? So have you ever heard, if you study in a university or a college, have you heard about shuttle buses? Are there any shuttle buses? This term, have you ever heard? Shuttle bus? No? Nobody has heard about this term, shuttle bus or shuttle? Yes. So what are shuttle bus? What does shuttle buses do? So they will from your department they will take you either to the main gate, and from the main gate they will take you to the department. So we have, if you have a very big university, then just like BHU or some other universities which are very big, so you have buses inside the campus that just takes you from various departments to your hostel, to the main gate, and like this. Those buses are shuttle buses. Shuttle bus because they shuttle between hostel to gate, to gate to hostel. That means to and fro, they are going from one point to other, then again coming from that point to the other point. They are just to and fro, moving from one place to another. That is shuttling. Shuttling means moving from one place to another place and then again coming back to the original place. That is shuttling. So similarly, the concept of shuttle, the word literal meaning of shuttle is this only. So if I take it in the context of proteins, so shuttle proteins are the proteins especially for transportation, that shuttles between, that moves to and fro between the nucleus and the cytoplasm. Nucleus cytoplasm, they are taking certain, so they are helping in the import or export. They help in import and export of certain RNA molecules, proteins, whatsoever. So Basically, when RNA is moving from the nucleus to the cytoplasm, when it is exported from the nucleus to the cytoplasm, various shuttling proteins are there which are helping it. So sometimes these shuttling proteins have to be removed so that transportation can be more rapid if their work is completed. So just like we discussed that RNA helicase can work either on RNA, double-stranded RNA or RNA protein complexes. So in similar way, it helps in the removal of shuttling proteins. Then RNA helicases in translation also RNA helicase works. So what it does is that intra-RNA interaction in mRNA. So basically, um, if this is your RNA, okay, and this is your intra-RNA interaction. If some inverted repeat sequences are there, if this sequence region is complementary to this region, then it can try to loop out itself. So this kind of looping out, it will create problem in translations. Okay, so this intra-RNA interaction is prevented with the help of RNA helicases. Then they also help in ribosome assembly and disassembly, and they also helps in the translation termination. So we have studied that what are RNA helicases, what are the different domains which are present in RNA helicases, and then what are the different functions of RNA helicases: splicing, maturation, export, and translation. And excuse me. Okay. And in detail, we have studied about the RNA helicase role in RNA export and RNA translation. Now, now comes the other important topic of this particular week is the nuclear pore complex. Now, what is the meaning of nuclear pore complex is that, see, I'm just giving you a gist, okay? I'm giving you a gist. See, what is needed in science is that you should understand the concept. What is the concept and what is the logic behind a particular topic? If you are clear with the concept and the logic, then you can go in depth. Going in depth without knowing the basics is of no use. So I will try to clear your basics, because if I am able to clear your basics, then whatever extra information that sir has told in his recorded lectures, that you can easily understand. But if you do not even know the basics, then all those extra in-depth knowledge will go above your head. So I'm trying to clear your basics and then we'll eventually solve questions also. Fine. So when I talk about nuclear pore complex, this is your cell, suppose, and this is your nucleus. So whatever has to, and this is your nucleoplasm and this is your cytoplasm. So RNA is synthesized inside the nucleus. Here RNA is synthesized. After the synthesis and maturation, this RNA has to go out in the cytoplasm, and here only translation will take place. Transcription takes place here and translation takes place in the cytoplasm. Now the question comes that how this RNA can be exported out from the nucleus to the cytoplasm. Okay, we have read that RNA has 5 prime cap, and 5 prime cap has some signals, and those signals kind of help in the transportation of RNA from the nucleus to the cytoplasm. Okay, we have studied this. How it will cross the nuclear membrane. This nucleus has a nuclear membrane, no? So how this RNA can cross the nuclear membrane? That is one important question that should come into your mind. So this RNA can cross the nucleus, can cross the nuclear membrane, just like we cross or we go from one room to another. How we go from one room to another? If we have walls, we go through doors, right? Because in between two rooms, if you have walls on all the four sides, then you will have a small door, and that door, you, with the help of that door, you will go from one room to another, or with the help of that door, you will go from your home to outside. Similarly, for RNA to go from nucleus to the cytoplasm, they have doors in the form of nuclear pores. Okay, so nuclear pores are nothing but like doors that we have in our homes. So those nuclear pores, it looks so simple that okay, nuclear pores are there, some openings are there, RNA will go from through those nuclear pores, it will go outside the cytoplasm. But no, it it has so complex and detailed machinery of how an RNA will go from the nuclear pore to the cytoplasm, how it will get the energy, how it will there be any protein molecules that will help in its movement, import and export. So that is a separate concept, and that is a separate dedicated lecture to study, that how RNA can be transported from the nucleus to the cytoplasm through the help of nuclear pores. And these nuclear pores have a very detailed machinery, very detailed architecture, very detailed infrastructure, which is called as nuclear pore complex. Okay, excuse me. Okay, so just a small pore, if we look through a microscope in a magnified manner, looks like this, which looks kind of scary though. Anyways, so nuclear membrane is a double layer membrane. You have a outer membrane and you have an inner membrane. Okay, and between the outer and the inner membrane you have a lumen. This is the nucleoplasm, this is the cytoplasm. So this is something like this. This is the nucleus. Okay, this is the nucleus and this is the pore. This is the inner membrane. This is the, sorry for the band handwriting, I'm just trying to explain you. This is the outer membrane, this is the cytoplasm, this is the nucleus, and in between you have lumen. Okay, so and this is your pore. So you can see that this pore has so many different kinds of things. It has a central framework, it has a channel in between, it has some filaments. Filaments which are inside the nucleus are known as intranuclear filament, and filaments which are at the cytoplasmic level, they are known as cytoplasmic filaments. Now, not only RNA, many other things, it can go outside to the cytoplasm also, it can be exported to the cytoplasm also, and things can come inside also, so it can be imported also. So there are two things, first is nuclear export and the other one is nuclear import. Okay, now this is important. So nuclear export requires nuclear export signals, and it requires nuclear localization signals. So if you have to export means giving it outside, bahar bhejna, it is giving you outside. So for RNA to go out from the nucleus to the cytoplasm, it needs nuclear export signal. For something to come inside the nucleus, it needs nuclear localization signals. And then the receptors for this are karyophyrin exportins. Here the receptors are importins. Both export and import requires the activity of GTPase, RAN. And here in the cytoplasm we have RAN GDP. Cytoplasm we have RAN GDP. In the nucleus we have RAN GTP. Okay, and then here what is needed is RAN gap. Gap means GTPase activating domain. So in the for export we need RAN gap, for import we need RAN GEF, that is GDP exchange factor, and this is the cargo which is responsible for exporting from the nucleus to the cytoplasm. This is the cargo importing, and then these are the things which are released. You just have to read about this. This is I'm just giving you a basic. You have to go in depth to read about it. Okay, I cannot cover each and everything because already it's 1 hour. We have to cover other things, and plus we have to solve questions also. Then just a basic diagram to show how things happens. So cargo, whenever you in cellular in, so basically this part, this portion, if you want to read from somewhere, then this portion comes under cellular biology. Okay. uh, yes, anybody wants to say something? Okay, so cargo means, just like cargo is a generic word in English. Cargo means all the luggage, all the luggage that has to be transferred. Okay, so here also cargo means whatever protein, whatever RNA, whatever the things that has to be transferred in and out of the nucleus. So this is the cargo. Okay, this is the cargo. Now what happens, that this cargo, and this is the FG repeat domain. These yellow ones are the FG repeat domains. This orange one is the cargo. This is the receptor through which the cargo will attach. So what happens? This is the cargo. It will attach to the receptor. Okay. Now, after attaching to these FG repeats are all, see, so many FG repeats are there. These FG repeats are very important actually, because these repeats actually help in the transportation. Then you see, this is the cargo and this is the receptor, and FG repeat is also there. Then it will go and it will bind to this FG repeat. This is the FG repeat. You can see this is the cytoplasm. So that means any protein is coming from the cytoplasm to the nucleus. So for protein or RNA, and it can be anything. Let's say it's a protein for our understanding. So first this protein will bind to the receptor, then it will bind to the FG repeat domain, and then it will form a complex. Now this will push itself interior, and then it will go again, it will join with some ran GTP. It will bind with ran GTP, and that ran GTP will provide the energy, and then this would be released from the complex, and then it goes inside the nucleus. This is how from the cytoplasm to the nucleus this protein is transferred. First it will bind to the receptor. After binding to the receptor, it will bind to the FG repeat domain. And after binding to the FG domain, RAN GTP will come. This GTP will provide energy, and then it will go inside the nucleus. Now we will solve two questions, and then we again move to other portion. So FG nuclear porins are located in nuclear membrane, nucleolus, nuclear pore complex, or cytoplasm. Tell me the correct answer. I'll give you 1 minute to think. Okay, I have to 1 minute to think, and then I'll switch the screen. Okay, let's see. Okay, Kavita has answered C, Jahanara has answered C, and Tejaswini has also answered C. What is C? Nuclear pore complex. Yes, you all are correct. Good. So, FG nucleoporins are located in the nuclear pore complex. Here only we have seen this is the nuclear pore complex. These small yellow ones, these are your FG proteins. Next question is, I don't know whether this portion has been completed up till now or not. Just a minute. Exportin. Okay. Just go through, before solving this question, I would like all of you to just go through this slide. So exporting cargo ran GTP complex, which is required for export. Importing cargo complex, which is required for import. Now, exporting T function depends on RNA quantity inside nucleus, ran GTP, ran gap, exporting 5. Excuse me. 1 minute to think, and then I will switch the screen.
RAN gap.
RAN gap. Okay. Let me see if others have also answered something. Nobody has answered? Okay, no, no. Uh, B, B, and C. C. Let's see. B, B, and C. Okay. Okay. There's one question also. I'll just tell you about that also. First, we'll do this question and then we'll come. Now, you see here, exporting cargo, it require RAN GTP complex in the nucleus. Here it is written. Okay. So, exporting cargo require RAN GTP in the nucleus. So here it is written exporting function depends upon. So the answer is RAN GTP. So this table is very important. Okay. So I'll just for 1 minute just go through this table because this table is very important. Afterwards, whatever we have to study, it depends on this table only. Just for 1 minute, go through this table.
Ma'am, there is RAN gap also.
Hmm?
Exporting,
RAN,
there is RAN gap also.
yeah, there is RAN gap also. But here, what the question is written, um,
Depends on.
actually, the see where it is, huh? So the RAN GTP is also required, and RAN GTP is also required. RAN gap, where you are seeing RAN gap,
Second point.
huh? So, but the question, I think the question you cannot, you have not understood, or by the language is not very proper. Basically they are saying that what is the cargo which is responsible in the nucleus, and there is a cargo which is released in the cytoplasm. Okay, so these all points are written does not means that these all points have same function, or they perform at the same time, or they have, like, in the whole process they have different functions at different time points. Okay, so this is just RAN gap means what? So you have RAN GDP in the cytoplasm. This is your cytoplasm. There is GDP here. That means RAN gap is acting on this RAN GDP. GDP. Okay. You have to understand this. Gap means what? Gap means GTPA's activating domain. That means it will activate GTPAs, and when this GTPAs is activated, GTP is converted into GDP. Okay. GT GTP is converted into GDP because of this GTPase activation domain RAN gap. Because in the cytoplasm RAN GDP is there, this RAN GDP is because RAN gap is there. Okay, RAN gap is actually helping in the hydrolysis of GTP into GDP. Whereas in the nucleus, you have GTP because you have this GDP exchange guanine exchange nucleotide factor, which helps in the exchange and reassociation of one phosphate group with this, so that it again becomes GDP. This happens because of GEF. So RAN gap is written over here, but that, I mean, this is just to show the points. The context in which we are talking in this question, that is entirely different. Here we're talking about the exporting function. So exporting function depends on RAN GTP, whereas RAN gap is responsible for conversion of GTP into GDP. Just mere presence of that point doesn't mean that it will, the answer would be this thing. C. Is it clear? Is it clear? Yes or no?
Yes,
Yes.
ma'am. Yes, ma'am.
Now someone has asked me what are FG proteins. Okay, so where it is. After this, I'll move a little quick because we have to complete a lot of things. So FG proteins are nothing but F stands for what? In this thing, protein nomenclature, F stands for phenylalanine and G stands for glycine. So basically, FG are phenylalanine and glycine repeats. Okay, and these are hydrophilic amino acids. Both are hydrophilic amino acids, and these are the hydrophilic amino acid stretches, and they are very flexible. You can see they can be folded very easily here at all the points. They can be folded very easily. They are very flexible, and they help in the import of the import and export of the cargo. That is the function of FG repeat domain. These are basically domains which are present in the nuclear pore complex. FG stands for phenylalanine and glycine. Okay, now I have to move a little quick because we have a lot of thing to cover. Now I think your RAN GTP and GTP point would be clear. So you have to go through this RAN. Now we come to lecture 3, RAN GTPase cycle. So just like I said, that inside the site, this is cytoplasm and this is nucleus. Okay, now in the cytoplasm, your GDP cytoplasm, in cytoplasm, GDP is more. In nucleus, I mean in the nucleoplasm, GTP is more. Okay, so this, if we have gone through the chapter, through the online class, through the recorded lectures also, so this RAN GDP is more, that is why it is bigger. RAN GDP is less, therefore this is smaller. So this is a cycle, so we can start from anywhere. Suppose uh, from where should I start? Okay, from anywhere I will start. So this is your RAN GTP. So RAN GTP means triphosphate. GTP means guanosine triphosphate. It means 3 phosphate groups are there. So when one phosphate group is removed, it is converted into RAN GDP. And this RAN GDP helps in the import of whatever things are there into the nucleus. And then inside again, this RAN GDP with the help of guanine nucleoside exchange factor GEF, this GDP would be replaced by GTP, and then again it will form RAN GTP, and then again RAN GTP, when it will cross the the membrane, it will go to the cytoplasm again because of the gap. So here gap is there and here GEF is there. So gap will remove one phosphate. So triphosphate would be converted into diphosphate, DP. It will come to the nucleus. Again, because of the nucleoside, I mean GEF, it will again, this GDP will be exchanged with the help of GTP. This is the RAN GTPase cycle which occurs. Okay. Now I have to move a little quick. So these are just some of the screenshots I have put. You can just go and read through it. I have just kept it so that you can read about it if you're not able to find them. Now again we come to lecture 4, where we're talking about nucleoporins and miRNAs, that is microRNAs. So basically nucleoporins are, what are nucleoporins? Basically they are formed by different nuclear proteins. Okay, and these nucleoporins are three kinds or of three types. First is FG nucleoproteins, which are nothing but phenyl and glycine rich repeat, that you were asking what are FG repeats. So there are three classes of nucleoporins. So nucleoporins are nothing but different combinations of nuclear proteins. Okay, different nuclear proteins combined together to form nuclear porins, and these nucleoporins are of three types. First class is FG nucleoprotein, second is nucleoporins, and third is NUPs. So this nucleoporins are nothing but phenylglycine repeats, and this is nucleoporins, and other one is NUPs. These are the three kinds of nucleoporins which are responsible for the transport of RNA. Then this is normal RNA transport that we have talked about. Then there is, specifically we will talk about tRNA transport. So how tRNA is transport? Same, export and import in same process is there. The only thing is that the receptor which is used for tRNA transport is exportin T. This is the receptor which uses RAN, which works in RAN GTP manner. Okay, so basically this question, although in general also it is required, exportin T functions, this is basically for tRNA transport. But in general also, if we're talking about exportin function, it requires RAN GTP. So here it is written. So exportin T is the receptor and it functions in RAN GTP dependent manner. Okay. So what happens? Ki first, uh, export means it has to go from the nucleus to the cytoplasm. Okay, from the nucleus to the cytoplasm it has to go. So what happens is that first it will bind to its receptor, which is exportin T. The tRNA will bind to its receptor, which is exportin T, and then in a RAN GTP dependent manner it will be taken out of the nucleus. So how it happens is that this exportin T RAN GTP, this whole complex would be formed. This whole complex will reach the cytoplasm, and as I told you, here RAN gap will stimulate the GTP hydrolysis, and this GTP would be removed, and then this tRNA would be exported. So what happens? This is your tRNA. I'm just explaining you one transport mechanism, and then you can understand all by yourself because all are similar only. tRNA, it will bind to exportin 1, which is its receptor. Now we have tRNA exportin 1 complex. tRNA exportin 1 complex. Now this complex has to go out of the nucleus, so obviously it needs some energy. It combined with RAN GTP. Now this RAN GTP kinds of hold the hand of tRNA exportin 1 complex and it takes it to the cytoplasm. Now in cytoplasm we have a lot of gap proteins, GTPase activating protein. In cytoplasm we have gap. So what it will do? It will hydrolyze and RAN GTP would be converted to GDP and it will detach from the complex, and hence your tRNA, it goes to the cytoplasm. So in this way tRNA is exported with the help of the receptor exportin T. So is it now clear that why exportin T has RAN GTP function and not RAN gap function? So the
Yes.
one who, is it clear now?
Yes, ma'am.
Okay. So this much detail I won't be giving to all the mechanisms because then we won't be able to finish the class. But you just have to understand the basic mechanism is that whatever is your, whether it's an RNA or it's a protein, it will first bind to the receptor, and then this complex will bind to some GDP mechanism, whatever, depending on whether it is in the nucleus or in the cytoplasm, and then with the help of some energy it will be transported to the nucleus or cytoplasm wherever it is going, and then the complex will detach itself, it will be broken down. Then this tRNA transport can happen with the help of exportin 1, and there is another receptor also which is known as loss 1, which is an ortholog of exportin 3. So this is the general pathway, and there is also an alternate pathway through which tRNA can be exported. Then now we come to the biogenesis of microRNAs, how microRNAs are formed. So basically you have to understand that mRNAs and microRNAs this is, you can just read over here. I'm uh, giving more attention to how miRNAs are formed. So basically what happens, that this is a stem loop structure. This is known as a stem loop structure. Now in this stem loop structure, some RNase 3, which is known as drosha. So this RNase drosha, it acts on this stem loop structure with combination with DGCR8. So the stem loop structure is cut in the nucleus by RNase 3. So basically, which is DROSA in complex with DGCR8. So how miRNAs are formed? That miRNA is nothing but a RNA which is present in a stem loop structure. MicroRNA is nothing but a single stranded RNA which is present in a stem loop structure, which is acted upon by RNase 3, which is known as DROSA, in combination with DGCR8. This cut will produce a 65 nucleotide stem loop intermediate, which is known as pre-miRNA. Okay. Then this pre-miRNA, it goes to the cytoplasm with the help of RAN-GTP, just like we have discussed. In the cytoplasm, gap will hydrolyze, and then this pre-mRNA is released in the cytoplasm. Then dicer, again it acts on the pre-miRNA and it converts it into a 22 nucleotide complex. miRNA duplex is formed. So this is how it happens. Let's see from the figure. See, this is your primary microRNA. Okay, this is the primary miRNA gene from which primary microRNA is formed. This stem loop structure is acted by DROSA and DGCR8. DROSA is the RNase which cuts the RNA, and then it forms the pre-miRNA. First, DROSA will act in the nucleus, and then this pre-miRNA is transported to the cytoplasm in RAN-GTP manner. Now in the cytoplasm there is gap, so GTPA would be hydrolyzed, and then this pre-miRNA is again acted by another RNase which is known as dicer. So dicer will again cleave it and it will form a 22 nucleotide miRNA duplex. Now this miRNA duplex will now go to the RISC complex. In the RISC complex, when it is in the complex, it will perform its function. Either it will do the repression of translation or it will cleave the miRNA so it cannot proceed for a translation. Okay, so this is how miRNA is formed, exported and function. So basically you should understand that there are two kinds of RNase 3 which are acting. One is DROSA, the other one is dicer. DROSA is acting in the nucleus or cytoplasm? Tell me. DROSA is acting on nucleus or cytoplasm?
Nucleus.
Nucleus. DROSA is acting in nucleus and dicer acts on cytoplasm. After the final cleavage, 22 nucleotide product is formed, which is your microRNA, which do the repression, which inhibits the protein synthesis either by translational repression or by mRNA cleavage. Then we come to lecture 5, where we're talking about the export. Now we come to the RNA export mechanisms of spliceosome RNA. So spliceosome RNAs are U1, U2, U5. So these spliceosome RNAs, again the same mechanism is there, that they will bind to a receptor which is known as CRM, which is also a exportin 1 receptor, and then they would be you can just read, this is not a very complicated thing, and then they will just move out of the nucleus to the cytoplasm. You have to just read it. I cannot explain each and every mechanism because you just have to read. There is nothing very much to explain. You just have to read. You just need to know the basic mechanisms. So these are all the different mechanisms by which your spliceosome RNA can be exported. Again, your export of mRNA, then mRNA, how they are exported, again it will bind to some kinds of factors. And important thing about mRNA is that till now we have studied that whatever export was happening, till now we have studied whatever export was happening, it was happening in ran GTP dependent manner. Okay, but here when we talk about mRNA, that mRNA export does not depend on ran GTP dependent manner. It depends on another class of proteins which is known as FGNUPS, FG-NUPS proteins. This is important. Like we have studied for tRNA, for spliceosome RNA, that the export was happening in ran GTP dependent manner. Ran GTP was taking it outside the cytoplasm, outside the nucleus to the cytoplasm. In the cytoplasm, then ran GTP was getting degraded. But mRNA does not happens in ran GTP dependent manner. This is important point you should understand. Then again, export of mRNA is directional. That means it will not go from cytoplasm to the nucleus again. It only goes from the nucleus to the cytoplasm, mRNA. And then again, this directionality is determined by some factors such as DPP1, GLE1, and then again these factors, this GLV1, it stimulates ATPase activity. This is nothing, you just have to read about it. Okay, these points I have just kept them in slides so that you can read it later on. Then again, after that there is rRNA export and rRNA import is there, or it's just export is there. Okay, then again you have rRNA export. Again you will have a receptor, it will bind to the receptor. Then there would be some again ran GTP would be used for hydrolysis, and then it will again go to the cytoplasm. So you just have to remember that details are there, that here you can see that there is an additional receptor is also there, which is MEX67 MTR2. This is additional receptor. Then there is an other export factor ARX1, which is also required. So the basic concept remains the same, that first your RNA will bind to the receptor, and then with the sorry, and then with the help of ran GTP complex, it will go to the cytoplasm. GTP will be hydrolyzed to GTP, and like this way it will go to the cytoplasm. Same is there, but little bit different details are there in other options. Then in lecture 6 what we have studied, that what are the diseases that can happen if this export and import machinery is disturbed. Just like we discussed that if splicing is disturbed, then there are different kinds of diseases that can take place. Similarly, if export and import of RNA is disturbed, it can lead to several kinds of diseases. One of the most common diseases that it can lead to is osteogenesis imperfecta, which is associated with aberrant mRNA export. That means the export of mRNA from the nucleus to the cytoplasm is not controlled. And why it happens? It happens because of a splice site specific mutation. There is a mutation in the splice site. Okay, there is a mutation in the splice site because of which this disease happens. Now we come to RNA quality control. Just like any process that we deal, whether it's DNA replication, transcription, translation, or in any industries also, you see there is always a quality control mechanism which takes place. So you can see when transcription happens, after transcription capping happens. Here also quality control takes place. That means first they will check that transcription is proper or not. If transcription is not proper, RNA would be degraded. Again, from capping to splicing, if capping is not proper, degradation will take place. From splicing to polyadenylation, if it is not proper, degradation. Polyadenylation to export, not proper, degradation. Export to translation, not proper, degradation. Translation is not proper, degradation. So you can see that so precisely each and every step is controlled and regulated, so that so thus the body or the cell tries its best
that there is no mutation that is there which can lead to some kind of disease or pathological manifestations. Then then if there is quality control, that means degradation of RNA also takes place for those RNAs which are not good or which has not passed the quality control test. For example, if this transcribed RNA has not passed its quality control test, it will be degraded. This RNA, if it is not spliced properly, it will be degraded. So that means that apart from the biogenesis of microRNAs, RNA production, transcription, RNA processing, post processing regulations, import and export of RNA, there is one process, one phenomena which is known as RNA decay. It also takes place. So, what is RNA decay? Cell need to remove the mRNA that are damaged or not needed anymore. Okay, any RNA which is damaged or which is not needed, it is decayed. mRNA decay pathways are found both in nucleus and cytoplasm. That means in both nucleus as well as in cytoplasm. In both, this decay will happen. mRNA have two stability determinants. That means the 5 prime cap and 3 prime poly A tail. So this is your mRNA. To find out whether this mRNA is stable or not, there are two determinants. First is 5 prime capping and other one is 3 prime tailing. These are the two stability determinants of mRNA. Now, decay can start only if one of the two structures are removed or there is endonucleatic cleavage. That means how the system or how the cell will know that this RNA has to be marked for decay or this RNA has to be decayed or destroyed. RNA, the system senses or the cell senses whether this RNA has to be decayed or not because of the presence or absence of these two markers. If 5 prime capping or poly A tail, either of them is not there, either of them is removed or either of them is not proper, then that RNA, then mRNA is marked for decay. Or if there is any attack on the mRNA within the, I mean, endonucleatic attack on the mRNA, then the system will know that this RNA has to be decayed. So there are two determinants, 5 prime capping and poly A tailing, that determines the stability of RNA. If either of them is removed, that means the RNA is prone to decay now. Or if there is any endonucleatic attack, then that will tell that the RNA is prone to decay. Okay, so now we'll solve the questions. So my humble request to everyone that I have put these screenshots just for your understanding, okay? Because it is beyond the capacity of the time to explain each and every point in detail in this class. It will take 6 7 hours for me to explain each and every mechanism. But as I told you, that the logic remains the same. If you know the logic, that first it is binding to receptor, then with the help of ran GTP complex it is going out, and then in the cytoplasm, as GTP is activating protein, that is gap is more, so it will cleave GTP into GTP, so GTP will be removed and the RNA is in the cytoplasm. This is the basic logic. Then you have some extra proteins, extra factors here and there in every different kinds of RNA that you can study. Now we'll come to questions. miRNA maturation is dependent on DROSHA, DGCR8 dicer or risk. So what is the answer? I'll give you 1 minute to think.
Dyson.
Yes. So let me see if others have also answered it. C. Yes. So the correct answer is dicer. So miRNA maturation, because uh, When dicer acts, then only the actual 22 nucleotide miRNA is formed. When Rosha acts, then the stem loop is converted into a 65 nucleotide structure. But when dicer acts, then actually the matured miRNA, which is of 22 nucleotide, is formed. That means the maturation of miRNA depends on dicer. Then next question is a disease that is the result if defective mRNA transport is there is osteogenesis imperfecta, osteomalacia, osteoporosis,
Option
osteomyelitis.
A,
Option?
osteogenesis imperfecta.
Yes. So then Jahanara, Renuka and others have also answered it correctly that it is option A. That is osteogenesis imperfecta. We have talked about that a mutation in splicing factor happens. Now, RNA undergoes decay. Why? Because it lacks coding sequence, it has stop codon, it has intron, or it lacks essential stability features. 1 minute to think and then I will switch the screen.
Option B,
Option D, okay. Any other, someone else who wants to try? We have one option D. Okay. So yes, option D is the correct answer. It lacks essential stability features. So just now we have talked about that the two stability determinants in mRNAs are the 5' capping and 3' poly A tail. If either of them is removed or there is an endonucleotic cleavage, then that mRNA is marked for delay. See, decay can start only if one of the two structures, that means 5' cap or 3' poly A tail, are removed or there is an endonucleotic attack. So the question is asking that RNA undergoes decay because it lacks essential stability features. And what are these stability features? This is 5' cap and 3' poly A tail. Okay. Next question is length of poly A on mRNA tail indicates that mRNA is undergoing decay, that mRNA is ready for translation, age of the mRNA, or nuclear location.
option B,
Option B. Okay. Others who wants to give a try? Janara has also said option B. Okay. I think this point we have discussed in very earlier classes. I don't remember. I think in second or third class only we have discussed this. When we were studying about the different post-transcriptional events that takes place, that time only we have discussed. I give you 1 minute. So, okay. So, actually the correct answer is C, that is age of mRNA. If you remember in the initial classes, we have discussed that the length of the poly A mRNA tail indicates the age of mRNA. That means as mRNA starts getting old, what happens? The length of poly A tail it also kind of increases. Okay, so this you have to always remember that length of poly A tail indicates the age of mRNA. Okay, so now we'll come to this question. Just solve this question. The proteins that are imported have nuclear export signal, nuclear localization signal, FG rich domain, or it has to be less than 55 kilodalton. Just solve this question. I'm going to get some water for myself. Solve this question. In 1 minute, I'll ask.
Option B.
So what do you think is the answer? Has anybody? Okay. Nuclear localization signal. Okay, and others? Jahanara has said C. B. Okay, so can you tell me what is the difference between nuclear export signal and nuclear localization signal? What is the difference between the two signals? When are these two signals used? Nuclear export signal, nuclear export signal, and nuclear localization signal. Export signal means these signals are needed to when? To move things outside the nucleus or inside the nucleus?
Outside the
Outside
nucleus.
the nucleus.
Outside the nucleus. So, nuclear export signal are the signals which are needed to uh, transport your RNA outside the nucleus. And nuclear localization signal means that these signals helps in the localization of RNA into the nucleus. Localization of RNA into the nucleus. So, import means here you have to understand whenever we talk about import. Excuse me, that whenever we're talking about import or export, we are talking in context to nucleus, that something going outside the nucleus is export, something coming inside the nucleus is import. So proteins are imported means by default it should come in your mind that it is going inside the nucleus. Okay, because this might be confusing, that if I what is the meaning of import, it is in context with the nucleus. That means the signals which are needed are nuclear localization signals. And what is FG rich domain, why it is where it is used? FG rich domain, where it is present? It is present where? FG rich domain, we have just learned.
Nuclear protein complex.
Nuclear
Yes, nuclear pore complex. Yes.
nuclear
So
pore
FG rich.
complex.
Yes. So good, Jyoti and Tarikshwari. I'm sorry if again I'm pronouncing your name incorrectly. So these FG rich domains are present in the nuclear pore complexes, which are basically phenylalanine and glycine, and which are required for the import or export of your RNA or protein, whatever it is. Now, nucleoporins are not important in RNA transport. True or false? Nucleoporins are not important in RNA transport. True or false?
False. False.
Yes, it is false. Nucleoproteins are very much important. Let us just revise that slide where we have studied about nucleoporins. Uh, hmm. So, nucleoporins, the heading itself says that nucleoporins are involved in RNA transport, RNA export. So, basically, your nucleus has nucleopore, and nucleopore has various proteins, nuclear proteins, and these nuclear proteins are nuclear pore proteins are present, are known as nucleoporins. So you should understand that nucleoporins are nothing but the proteins that are present in the nuclear pores. That is why they are known as nucleo, that means in the nucleus, and pore means in the porins, means in the pores. That means proteins which are present in the nucleus pores. And these nucleoproteins are of three classes. First is Fg nucleoproteins, second one are known as nucleoporins only, and the third is NUPs. So Fg nucleoproteins are the proteins which has phenylalanine glycine rich domain. Nucleoporins do not have phenylalanine glycine rich domain, and NUPs are the third class. And basically, these NUPs are responsible for the nuclear pore complex to to remain attached. That means if this is your nuclear membrane, this is your nuclear pore complex. So these proteins, NUPs proteins, they kind of help in the attachment of this nuclear pore complex with the nuclear membrane, so that this nuclear pore complex does not move anywhere. Okay, so these are your nuclear proteins, and they are helpful in the export also. They are, in general, they are helpful in transportation. Okay, three classes: phenyl glycine nucleoproteins that have phenyl glycine repeats, nucleoporins that do not have phenyl glycine repeats, and NUPs which are responsible to anchor the nuclear pore complex in the nuclear membrane. So that means these things that we have studied, uh, where it went, the slide where I showed you. Okay, the slide where we have studied about the Fg proteins, those are nothing here. So these Fg proteins, Fg mediated domain, they are nothing but nucleoporins. Okay. Next question is, so the answer is false. All RNA species follow same pathway of RNA export. True or false?
False.
False. Others? Janara, Jyoti. Okay. So why it is false? See, all RNA species means we have tRNA, mRNA, snRNA, and we have seen so many different mechanisms are there. And also, excuse me, and also one very important point to note here is that in general we have talked about that for the export of RNA from the nucleus to the cytoplasm, we need RAN GTP. Okay, but this RAN GTP is not used for the export of mRNA. mRNA is an exception. Always remember this. mRNA is an exception. Here it was written clearly, just to remind you. See, the first, in any export or import process, the first thing is that your RNA will bind to a receptor. Okay, now that export receptor, it is the requirement of the export receptor to take the RAN GTP. In every case, export receptor, whether it is exportin 1, exportin 5, exportin 7, whatever, the receptor requires the need of RAN GTP. But here, the receptor does not depend on RAN GTP. Otherwise, uh, 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 but it depends on a discrete class of proteins which are known as FG NUPs. Okay, this is one difference you should always remember. So from here itself we can say that the all the RNA species, they do not follow the same pathway for RNA export. So it is false. Okay, next question is, exosomes persistently degrade RNA indiscriminately. True or false? This is just in general, if you know, we have not discussed it, but if you have read the read about the classes, then you might have a little bit of idea.
True.
Okay, others also, if you want to try. So Janara is saying true. Let's see what others have to say. Kavita is saying false. Okay. Others, if you want to try. Okay. Jyoti, if you want to try anything you think it is true or false. Lakshmi Kanta, Swapna, and who else are there? I see only some participants. Same names always comes. Monica, Kanchana, Kusuma, Renuka. Okay, Kanchana has said B. Jyoti has said false. Good. At least you are listening. That means. Otherwise, I was thinking that some of the students they just start the class and do their work, like mostly students do in online classes. Anyways, so yes, the answer is false. That means so first you have to understand. So this is your homework because we don't have that much of time that I can explain you about the exosomes function. If you really want to know what, you just read that what are exosomes. So anybody knows what are exosomes? So anybody knows little bit about exosomes? No. Okay, so basically exosomes are nothing. So we have a in eukaryotic cell we have endosomes. Okay, we have particular vesicles or compartments where degradation happens. So within those extracellular vesicles, extracellular compartments, we have exosomes, which are nothing but membrane bound vesicles. Okay, so if this is your endosome, within the endosomes you have exosomes, which are nothing but membrane bound vesicles, which are basically used for many different processes. They are used for degradation. They are also used for some kind of circulation in I mean some kind of transportation, delivery of certain kinds of lipids or proteins. In fact, a lot of research is now going on exosomes, that how exosomes function. So a lot of research has shown that microRNAs, so microRNAs, they deliver their function with the help of exosomes. That microRNAs are present inside the exosomes, and these exosomes are actually secretory vesicles. Okay, so these exosomes are secreted, and then in within the exosomes microRNAs are there, and they microRNA then performs its function. Especially in my masters I have done, my seniors used to do, because my work was on reproductive biology. So there my seniors used to work on exosomes only. So they have shown that microRNAs are present inside the exosomes, and then these exosomes they act on the blastocyst, and then it helps the blastocyst for its nurturing and its growth. So it has been shown. So exosomes, essentially we know that exosomes are required for delivery or they are required for degradation. But now research is showing that exosomes are, because they are secretory vesicles. So, and even artificially now what scientists are trying, that anything they want to secrete or they want to deliver at other places, artificially they are synthesizing exosomes. It is so fascinating. Artificially they are making exosomes, and whatever they have to put inside the exosome, they are keeping it inside the exosome, and that is you that they are using as a delivery vehicle. Just like plasmids we use for cloning, delivery vehicle, similarly exosomes are also used as delivery vehicle. So exosome is actually a very growing research area, not just for this course point of view. I'm telling you in general that exosome is a very vast growing research area. Anyways, now next and the last question for the day is, de-adenylation is essential for RNA stability. True or false?
False.
False.
False. So can anybody explain me why it is false?
Because poly-A-tail is essential.
Yes. So basically, we have studied good that the RNA stability in RNA decay, this thing. Yeah. So in RNA decay, we have studied. So you can understand from the questions itself that which portion is important, which portion is not important. Not important, I mean, which is more important. So this RNA decay is an important topic. And then the basic concept of importing and exporting is very much important. You should always know the in-depth portion, but you should know the names of these different proteins and different receptor names. But uh, as you can see from the question, they ask you logics rather than tell the name of the conserved NES containing domain that is recruited in RNA export. They will not ask like you have to rectify and tell the name of these uh, different proteins. You should know the concept and the logic behind it, right? Anyways, as we were discussing that there are two determinants for mRNA stability. First one is your 5' capping, and the other one is poly A tailing. So, if any of them is not there, then that RNA is marked for RNA decay. So, here the question is asking that, sorry, that deadenylation is essential for RNA stability. Then, no, it is not essential because poly A tail is needed. Poly A tail and 5' cap are the things which are needed for the stability of mRNA. Okay, so up till now, what all we have studied, let's quickly see through the slides. First, we have studied what are the mechanisms of intron gain. We have seen that there are four mechanisms essentially. First is intron transposition, transposome insertion, where transposome is inserted and it is converted into a spliceosomal intron. Then intron transferred to a paralogous gene, or a self-splicing intron which is converted into a spliceosomal intron. Then we have studied what are orthologs and paralogs. If genes are separated through duplication, they are called as paralogs. If through a speciation event, they are known as orthologs. Homologous means common ancestry. Then we have studied that RNA splicing is also regulated, and the regulation of RNA splicing occurs by various mechanisms such as phosphorylation, acetylation, and all of these. And you have a major spliceosome complex and minor spliceosome complex. Your minor spliceosome complex is just like a plan B. When major spliceosome complex is not working, then minor spliceosome complex comes into action, but both of them performs the same function. Then we have studied that what are the abnormalities or physiological pathological conditions that occurs if there are any defects in splicing. Defects can be of two types. Either there are mutations in the splicing factors itself, or the pattern of the splicing is changed. So these are the different diseases that are caused. Then we switch to RNA helicases. So RNA helicases are nothing but which are required for the unwinding of RNA, and for the unwinding processes they need energy. So that means they hydrolyze ATP mostly, and their substrate, that means the RNA helicase acts on either double stranded RNA or RNA protein complexes. And there are different RNA helicase families, and then these RNA helicase uh, have some roles in pre-mRNA splicing, maturation, RNA export, translation. Then we have studied in detail, sorry, we have studied in detail the role of RNA helicase in export and translation. So in export, we have studied it is responsible for the correct conformation, unpacking of RNA protein complexes, and removal of shuttling proteins. And then in translation also it is required for preventing this intra RNA interactions. Then we come to nuclear pore complex. We studied that the whatever your RNA is there, it has to go, it has to pass through this double. So it has to pass. Just a minute. It has to pass through this double layer membrane. So it passes the double layer membrane with the help of this nuclear pore complex. And there are two things. First is nuclear export and nuclear import. And then we saw that how it happens. The your RNA, it first bind to its receptor, transport receptor, and then it binds to the FG repeat domain. Then it goes inside, and then with the help of RAN GTP, it comes out of the nucleus to the cytoplasm. And then we saw the RAN GTP GTPase cycle. Always remember, in the cytoplasm, GDP is more. In the nucleoplasm, GTP is more. And if GDP is more, that means gap is there. If GDP is more, that means GEF is there. Important. GDP, gap. GTP, GEF. Sorry. then Then we have studied about nucleoporins. What are nucleoporins? What are the different classes of nucleoporins? Nucleoporins are nothing but the proteins which are present in the nuclear pore complexes. Then, just like we have discussed the basic mechanism of transport, then there are different kinds of transport happens for different kinds of RNAs. For tRNA, you have exportin 1, and then there is another receptor which is loss 1. Again, it requires uh, ran GTP activity for the export of tRNA. Then we studied the biogenesis of miRNA. For miRNA, we require exportin 5. And then we studied how miRNA is formed. So from the primary mRNA, this stem loop structure, DROSHA and DGRC acts on it. It converted into a 65 nucleotide structure. It goes out into the cytoplasm. Dicer acts. It is matured and formed a 22 nucleotide structure. Then we come to the export of small nucleolar RNA, which are nothing but your spliceosomal RNAs. And for spliceosomal RNA, the receptor is exportin 1. And some other points that you can just study. Then we come to the mRNA. Important point in mRNA is that RAN GTPA is not required. Otherwise, else you have FG NUPS which is required. Then you can read about rRNA export. For rRNA export, the receptor is CRM1, and there is additional receptor which is MEX67 MTR2, and there is additional export factor which is ARX1 which is required. Then these are the diseases which are linked. If the export and import is improper, important disease is osteogenesis imperfecta. And then we have studied about RNA decay, that the two important determinants of RNA stability are 5' capping and 3' poly A tail. If either of them is missing, or if there is any endonucleotic attack, then your RNA is prone to delay or decay. Okay, so with this we have completed today's lecture. I hope you were able to understand at least a little bit of it. So these things can be, it may look a little overwhelming. So many names of different proteins, different mechanisms for rRNA, tRNA, mRNA, everything is different. But the concept remains the same. The logic remains the same. First, RNA will bind to its receptor, and then receptor will take the help of RAN GTP. RAN GTP, with the help of RAN GTP, RNA, receptor, and RAN GTP, all three things together, it will move from the nucleus to the cytoplasm. In the cytoplasm, GTP is activating protein, that is gap proteins. It will act on GTP, and GTP would be released, and your uh, RNA would be released in the cytoplasm, except for the case of mRNA, where GTP is not used. Another set of proteins, NUP proteins, are used. So with this, we have completed today's class. So I hope you were able to understand something. I tried my best, considering my little bad voice and little illness. I hope I was able to deliver it properly. So thank you, everybody. We'll stop the class for today.
Okay, ma'am.
Thank you. So we'll meet. We'll meet next week now. Bye. Take care.
Bye, ma'am.